bioluminescence of the substrate, okay, that can glow in the dark, okay, and can be detected. The light can be detected by various system, okay. Okay, so this one is one example. Okay, you can read the notes down here. Okay, if you want to watch a video, also I have provided a YouTube here, but to explain to you in a simple and fast way. So we have the regulatory gene here in the light operon model, and this is a light operon comprised of like Z gene, like Y, and so like A gene. Okay, so the control region or the regulatory region, okay, will comprise of the like CI promoter, like CI gene, and we also have the like operon promoter and also the promoter, like operon promoter. Okay, this region, and then promoter, and then this one is the operon. Okay, and then after that, for this regular gene, when the metal is present, so the what happen is this like CI mRNA will be expressed, and it will also lead to the this like operon gene to be also expressed. Okay, so when this like mRNA is being expressed, so it will produce three enzymes: galactosidase, permease, and also transacetylase. And this, when it react to the substrate, it will produce color change, okay, on the medium, okay. And this for like mRNA, what it will produce, okay, is a repressor protein. So when the repressor is needed, when the the system doesn't detect the metal anymore, this repressor protein will stop the production of galactosidase, permease, and transacetylase, and it will repress the expression, and eventually will stop the production of this enzyme, and color change will be no more happen. Okay, so this is the controller or the regulatory gene that is needed, because this galactosidase, permease, and transacetylase will only be produced when the metal is present. And once the metal is no more present, it won't produce this enzyme anymore. The production will be stopped, and the repressor protein is important to stop the production of this particular enzymes. Okay, right. I hope you understand that one. Okay, then okay, this is just to recap. Okay, the system. Okay, you can see the one I mentioned earlier, and now we go to the biosorption, okay, tool for. It's a tool for environmental cleaning by absorption mechanism. Okay, it can be divided into, the first one is a metabolism dependent, and the second one is a non-metabolism dependent. Okay, this is also important for your exam. Please take note. Okay, so the metabolism dependent can be separated or can be identified as two processes. Okay, the first one is transport across cell membrane, and the second one is precipitation. The non-metabolism dependent also can be identified as precipitation. However, there are three more processes that happen, which is physical absorption, ion exchange, and also complexation. So these are the non-metabolism dependent, meaning the absorption no need to have the metabolism, meaning no cells are involved. Okay, for metabolism dependent will be involving the cell. Okay. Okay, so as I did mention to you just now, so metals by bioemission mechanism. First one is metal that will happen is metal immobilization. So how can this happen is through complexation, which is bioaccumulation, and also bioabsorption. Bioaccumulation is wrongly spelled like bioaccu, ACCU, accumulation. Okay. So how this complexation can happen through bioaccumulation, also properties by the microbes. So when the microbe produce azopolysaccharide, when I say exo, it is out at the outer part of the cell. So it's a polysaccharide that are being produced at the outer part of the cell, and so the lipoprotein also will be produced on the surface of the cell, and this will lead to the metal to be complex, okay, on the surface of the cell, and as in certain part of the cell, it can start to be absorbed within the cell, but not go deeply in the cell, but just at the outer layer of the cell wall. Okay, it could also another way of metal immobilization is through precipitation. So for example, the hydrogen sulfide, okay, producing bacteria. So this bacteria can produce siderophores and also metal reduction enzymes. Okay, so what this when this H2S producing bacteria growing, so it will through this siderophores and so metal reduction enzyme, and so exudates. Okay, so it will precipitate the metal. And on another process, okay, we call it as solubilization, and this can be categorized as bioleaching. So how this bioleaching happen is through the production of organic acid, siderophores, and also root exudates, okay, that are being produced by the plant. For root exudates, plant siderophores can from bacteria, organic acid can be from bacteria also from the plant, okay, plant. Okay, and then okay, this one is just to give you the overview, okay, in the form of a schematic diagram, okay, how metal can be immobilized by the microbial biomass. Okay, so if you just regard this structure, the hexagon, orange one, okay, over molecular hexagon is metal, and this green spiky structure here or shape here is actually microbiome biomass. Okay, so how the metal is being organized by the microbiomass is this is the structure that happened. So the microbes will surround this hexagonal metal, and the microbes can be in the form of fungi, bacteria, yeast, and also algae. Okay, so when the microbes surround this particular metal, so we can see that this metal has been immobilized, okay, by the microbial biomass. Okay, right. And then for absorption, so if you want to know the difference between bioabsorbent and absorbent, okay, so absorption and bioabsorption. Okay, so absorption, okay, we have this one doesn't involve biological system, so it is physical and chemical. So the metal can be called as adsorbate, okay, adsorbate, while the absorbent will be in the form of membrane or solid substrate. Okay, here, so the metal attached to the membrane, and this is called as absorption. While for bioabsorption, okay, we call this metal as biosorbate because it now involves biological system, and the biosorbent is actually the membrane of living cells. Okay, for instance, bacteria, fungi, algae, and so biopolymers. Sometimes not necessarily it will be in the form of living cell. We can also extract these biopolymers from the living cell, and then we take out this living cell and make it into biopolymers. Okay, so without the living cells anymore. So, but because we derive this particular polymer from a living cell, so we call it as biopolymers. Okay, and then this is the attachment, okay, of the metal, that is, and because it involves the biological living cells, okay, or the biopolymers, so we call this process as bioabsorption. So what is bioabsorption? Okay, bioabsorption is a property of certain types of inactive or active organisms to bind, either inactive or active. As I did mention to you before, inactive in the form of what do you call that? Biopolymer. So this one is inactive. When active, meaning it's a living organism that can bind, okay, and also concentrate heavy metals from even a very dilute aqueous solution. So bioabsorbents can be classified into the first one, inactive organism, mainly include algae, fungi, and bacteria, and their derivatives, okay, which are termed as biopolymers. Opposite to bioabsorption is metabolically driven active bioconversion by living substances. Right. So I have to skip a few slides. Okay, this one you can read because otherwise I cannot complete it within the time given, because we have another set of lectures.